Hello and welcome everyone to the fifth Consciousness and Reality Colloquium for this academic year. I am Kunal Mooley, an astrophysicist and the co-founder of IMIX, which is the Institute of Mind, Intelligence and Consciousness Studies. Our participants today span several institutions across North America, including Caltech, MIT, Stanford, and universities in the UK, namely Cambridge and Oxford. So this CNR Colloquium series seeks to inspire interdisciplinary investigations on topics such as qualia, cognition, mind, consciousness, and the nature of reality. Some quick logistics before we introduce our esteemed colloquium speaker today. I request all of you to please note that there is a Q&A button at the bottom of your Zoom windows, and you can post your questions by clicking on that button, and selected questions from that Q&A box will be taken after the talk. And so it is a great privilege to introduce Biyu, who is an associate professor of neurology, neuroscience, and radiology at the New York University Grossman School of Medicine, where she also leads the Perception and Brain Dynamics Lab. Dr. Hay completed her PhD in neuroscience from Washington University in St. Louis. Before joining faculty position at NYU, she established her own independent research program at the National Institutes of Health. Dr. Hay is widely recognized for her pioneering work on the neural mechanisms of conscious perception. So it's going to be an exciting talk, but her research and works have provided critical insights into various important questions like spontaneous brain activity, one-shot perceptual learning, predictive processing, and so on. She has received numerous prestigious awards, including the Wilczek Prize for Creative Promise in Biomedical Sciences, the Irma Herschel Career Scientist Award, and also the Society for Neuroscience Trubatch Career Development Award. So please join me in welcoming Dr. Biyu. Please take it over.
Thank you very much for having me. Let me share my screen here. Do you see it okay?
Yes, perfectly fine.
Okay, great. All right. Thanks for having me. All right. So in honor of this seminar series, which is titled Consciousness and Reality, and some of the physicists among the audience here, I'd like to start with a quote from Albert Einstein. In this short essay called Physics and Reality, published in 1936, he said, "Physics treats directly only of sense experiences and of the understanding of their connection. But even the concept of the real external world of everyday thinking rests exclusively on sense impressions." I think what he means is that understanding sense experiences and consciousness would not only shed light on our lived human experiences, but also have implications for our understanding about the external world. As Kunai mentioned, I'm interested in how the brain generates conscious awareness. And my lab takes visual perception as our model system. So our goal is to understand in this line of work how the human brain generates vivid visual experiences, such as seeing the redness of red, seeing the beautiful sunset. The classic neuroscience textbook picture of how visual perception happens is very much centered on the feed-forward pathway in the brain. What we mean by that is this one-way information flow from the light hitting the eye to that information being transmitted to the thalamus, the visual part of the thalamus in the brain, and then to the first stage of cortical processing, the primary visual cortex, and forward onto additional later visual regions. We have a wealth of foundational important neuroscientific knowledge about how visual neurons at different stages of this feed-forward pathway function and what their tuning properties are. For example, we know that neurons in primary visual cortex tune to edges and low-level features, and intermediate regions might tune to shapes, and a region in the end stage of the ventral visual pathway called inferior temporal visual cortex, the neurons might tune to high-level properties such as categories. So this feed-forward mechanism, this framework, has been very powerful in explaining how the visual system generates invariance to different changes in low-level visual properties and allow us to identify and categorize visual inputs such as a tree, a chair. And its power has been mirrored by the successes of deep convolutional neural networks, which also are completely feed-forward once they are trained. And units at different stages of deep CNNs perform this sort of a canonical linear nonlinear functions that mirror the neurons in the brain. So it's a very powerful and successful framework. The work in my lab over the last 10 years has shown that this feed-forward framework, while powerful, especially in explaining categorization, identification kind of behavior, some of the visually guided behaviors, it's not sufficient for explaining the neurobiological mechanisms underlying our conscious visual experience. So we have discovered that to explain the neural mechanisms of visual awareness, we need 3 additional ingredients that are outside of this feed-forward framework. One of them is intrinsic spontaneous brain activity that is constantly ongoing. And the second one is top-down feedback, which has a massive presence in the brain. And the last one is memories. Memories are shaping our perception all the time without us noticing it. So first, let me share with you some work we've done in elucidating intrinsic spontaneous brain activities' influence on visual awareness. This visual illusion illustrates this point. So this is a static picture, but depending on how far you're sitting from your computer screen, it probably looks like it's moving. So this moving sensation, this experience is generated by our brain's intrinsic activity, and we see a moving impression even though the stimulus is completely constant. And through the last 2 to 3 decades of systems and neuroimaging, system neuroscience and neuroimaging research, we have known that the brain has this large magnitude, energetically expansive, spontaneous activity that's constantly ongoing. The human brain accounts for 2% of the body weight, but 20% of our energy expenditure. And the vast majority, over 90% of that energy expenditure is used for maintaining this constantly ongoing spontaneous activity, which we know is richly structured spatially and temporally. And when this structure is disrupted, it can lead to behavioral and clinical deficits. However, we still lack a coherent understanding of how this spontaneous activity, which is constantly ongoing, impacts and shapes the brain's processing of incoming sensory stimuli and task demands. Put simply, the brain is not like a computer that is silently waiting for inputs and task instructions to arrive. It's constantly generating its own activity and, if you will, its own reality. So our way of thinking about how the brain processes external sensory input is to move away from this model of throwing a pebble into a peaceful pond, but into this picture of throwing a pebble into a roaring ocean. So in this first series of work I'd like to share with you, we asked how spontaneous brain activity that occurs in the brain and results in this constantly changing brain state, how does it impact whether a piece of sensory information accesses conscious awareness. So more than 10 years ago, we did this very simple experiment. We presented healthy young human volunteers with these so-called Gabor patches. It's an oriented patch, a very low-level visual stimulus. And it was briefly presented with its contrast titrated. So it's a very faint, brief visual stimulus. And when the identical stimulus is repeatedly presented to the same human participant, it is sometimes consciously perceived and sometimes not. So this allows us to ask how pre-stimulus brain activity predicts whether that stimulus enters conscious awareness. So on every trial, we asked the participant to both give us an objective performance, which is to categorize the orientation of this patch. You see pointing to the upper left or upper right. And if they didn't see the stimulus, just give us a false guess. Just guess it. And then we ask them subjectively, did you see this stimulus? And so we asked for an honest, truthful, subjective report. There was no right or wrong here. Just tell us if you saw it. And what we found was that when people saw the stimulus, their orientation discrimination performance was near ceiling, so about always correct. But even when they said, I didn't see the stimulus, their false discrimination performance was still slightly above chance level. This reproduces a pattern that has been shown by many previous behavior studies. So we recorded brain activity, dynamical brain activity with magnetoencephalography, which is kind of a fancy cousin of EEG that records brain waves when you go to the ER, if you need to have a brainwave recording for any reason. The MEG has better spatial resolution because the skull and scalp are transparent to the magnetic field. We were able to record dynamical brain activity from all these sensors, so hundreds of sensors covering the scalp. And from each sensor, the brain waves recorded follow the power spectrum that looks like this. So basically, there's a 1/f spectrum. So this is log power against log frequency. We see the ubiquitous 1/f power law distribution of the power spectrum. And on top of this, there are peaks at different frequencies that correspond to different brain oscillations, like the famous alpha oscillation or beta oscillation. So what we have found in this study and many other studies is that the low frequency component of the 1/f brain activity, which I have studied early on in my career before I started working on visual awareness. So the low frequency component of this 1/f activity, which we and others call the slow cortical potentials, allow us to decode at a trial-to-trial level whether the stimulus enters awareness. And in this study, we found that it even allowed us to predict whether that conscious access is successful even before the stimulus was shown to the subject. So let me show you what we found here. So on every trial, we take the MEG activity across the scalp at every time point with millisecond precision. At every time point, either before the stimulus input was presented or after the stimulus input was presented, to predict the subjective report given by the subject, seen versus unseen. Here we just used a linear multivariate classifier, so a very simple machine learning decoder. And what we found was that, neural activity recorded up to 2 seconds before the stimulus was even presented allowed us to predict significantly above chance level whether the subject was going to answer seeing the stimulus versus not seeing the stimulus. The purple bar at the top is where the decoding was significantly higher than the chance level of 50%. The stimulus, as I mentioned, was very brief, was only 66 milliseconds here. So this effect could be visualized beautifully in a low-dimensional state space. So here what we did was that we had 273 sensors covering the brain, and we projected this very high-dimensional brain activity state into a low-dimensional state by extracting the first 3 PCs, principal components. So this plot shows data from an example subject where the trials for seen trials, as the trials where the stimulus was consciously perceived, are color-coded in blue and red for the 2 different orientations. And the gray colors show the unseen trials. And the circles indicate when the stimulus is presented. And the brain states are plotted from about 1 second prior to the stimulus onset to 3 seconds after. So what you can see is that even before the stimulus input, the brain state is different. The brain resides in different regions of the state space for seen trials and unseen trials. And depending on that initial state, it travels on one trajectory corresponding to seeing the stimulus and a different trajectory corresponding to not seeing the stimulus. And we further found that in seen trials, there was this dramatic cross-trial variability reduction. And together, this suggests that neurodynamics underlying conscious perception could be described as initial state-dependent, robust, transient dynamics. So that was our initial foray into spontaneous brain activity's impact on conscious perception. More recently, we tried to decipher the spatial distribution of this activity in the brain using high-resolution, high-field MRI. So we ran a similar study in the MRI scanner where instead of using these ball patches, here we were interested in conscious perception of meaningful visual stimuli, such as pictures depicting animals, faces, or objects, or houses. And the task is very similar as before, where people first give a categorization report, which category is the stimulus, and then they tell us subjectively if they saw a meaningful object or not. Again, the stimuli were titrated to the recognition threshold so that when the identical stimulus is repeatedly presented, it's consciously perceived or consciously recognized in about 50% of the trials. So critically, we also included these face scrambled pictures, which do not have any meaningful content to get a sense of how likely a particular subject is going to answer yes versus no at baseline. So what's somebody's tendency to answer yes versus no? And here we found that even when the stimuli do not contain any meaningful objects, people say that they recognize a meaningful stimulus, a meaningful object in about 20 to 25% of the trials. We call those false perceptions or false alarms. And this paradigm allows us to leverage this powerful analytical framework developed by psychologists to analyze behavior data. So under this framework called signal detection framework signal detection theory, we consider the brain's response representation of a sensory input on each trial as a value along this internal representation axis. So noise stimuli would trigger a distribution that is more closer to the origin than a signal distribution. And we model them here with Gaussian distributions. And somebody's basic tendency to answer, yes, I see a stimulus versus no, I don't see a stimulus would be captured by this criterion, which if this criterion is high, it would mean that the subject is less likely to answer yes. And if it's low, they would be more likely to answer yes. So this allows us to capture sensitivity of a subject, which is how well they distinguish between signal versus noise, and criterion, which is how likely they are to answer yes. So we asked, how does pre-stimulus brain activity before the stimulus is even presented to the subject influence these two different metrics of conscious recognition? And here, using whole brain 7T, 7 Tesla MRI, which is a technique to record brain activity with very fine-grained spatial resolution, we found that there is a set of brain regions, including visual regions and prefrontal regions, where higher pre-stimulus activity predicted a higher criterion. So if these regions have higher activity prior to the onset of that visual stimulus, people tend to adopt a more conservative criterion. So they had fewer hits and fewer false alarms. And a different set of brain regions, insula and thalamus, and this anterior PCC region had the opposite pattern, where higher pre-stimulus activity predisposed the subject to adopt a more liberal criterion. And we think these regions are broadly part of the singular opercular network that's linked to arousal. And when we looked at pre-stimulus activity that influences sensitivity of conscious recognition, we found two specific regions in the midline. The MPFC, a very high order brain region, and the retrosplenian cortex, and there was a thalamus cluster as well. So in these regions, they invariably had a positive relationship to sensitivity, such that if the pre-stimulus activity was high, people's sensitivity in their conscious recognition was high as well. And VMPFC ended up being a critical node here that influences both sensitivity and criterion. In fact, this pattern predicts that if we contrast false perception trials with correct rejection trials, where we have a scrambled picture, it's either correctly categorized as not seeing something, correct rejections, or maybe it generates a false perception. When we contrast those, we find that VMPFC's activity is low prior to false perceptions. So we are testing this hypothesis currently about these regions' activity protects against false perceptions. So we looked into the mechanism here, and we found that when the MPFC spontaneous activity is high, it basically stabilizes sensory evoked responses so that both the noise distribution shown in gray and the signal distribution shown in green here have a narrower distribution. And that gives a higher sensitivity as well as a more conservative criterion in this threshold level paradigm. Whereas visual network had a different opposite effect on the variability of sensory evoked responses. When pre-stimulus visual activity was high, the evoked responses became noisier, and downstream decision-making regions adopted a more conservative criterion. And the singular opercular network regions, when their pre-stimulus activity is high, we found that there was a more liberal criterion and better categorization behavior, fitting with the idea that the transient arousal might be higher. But together, the big take-home picture from here is that we need to move away from thinking about the brain's processing of a sensory input as a serial steps of transformations, but move into more this interactive dynamical systems framework where sensory input interacts with the pre-existing brain state, which includes both the moment-to-moment ongoing brain activity, which we call intrinsic brain dynamics, as well as the activity-silent connectivity patterns as encoded in the synaptic connectivity, which I'll talk about in a little bit. Okay, so before I move on to the second part, I'd like to talk about this phenomenon of intrinsic brain activity generating conscious perception that is decoupled from the physical sensory input a little bit more. So there are these bistable pictures that have been known to psychologists for over a century. The pictures are ambiguous, such that even when you fixate your eyes at the center of the picture, what you consciously perceive fluctuates back and forth between the two different interpretations of the picture, such as a vase in the center or two faces facing each other, looking down on the cube or looking up at the cube. This one can be seen as a man playing a saxophone or a woman's face. This one can be seen as a duck or a rabbit. So using this bistable visual perception paradigm, we asked which brain activity allows us to predict at a given moment which conscious percept the person is experiencing. And it's a very wonderful paradigm because the physical sensory input is unchanging. So conscious perception can be decoupled from the external physical sensory input. And we found that, again, if we extract the fluid cortical potentials, we could decode at a given moment in time which percept is experienced by the subject. So here 0 is right after a button press and 100 is right before the next button press. Because the different trials have different lengths, we had to resample them to plot all the different trials together. So 20 would be 20% into one trial. Whereas when we look at, for example, amplitude fluctuations of brain oscillations in the alpha and beta frequency range, we could not perform this decoding. Interestingly, we found that these oscillations have a different function in conscious perception. So if we extract the amplitude envelope of alpha oscillation, so alpha oscillation is the very first brain rhythm discovered when Hans Berger invented the EEG. It's a posterior brain region that increases in amplitude when you close your eyes. It's very reliable. And we found that the amplitude envelope of this alpha oscillation did not predict the content of conscious perception, but it predicted how long that percept will last. So this is just an illustrative example from a single subject where every dot is a single trial. And you see that when alpha amplitude goes up, that percept duration gets longer. And this is a big effect from, you see percept duration goes from 1 second to 10 seconds, and alpha oscillation amplitude tracks that. So it appears that we have discovered that there are separate frequency channels in the brain for encoding the content of conscious perception and maintaining the stability of conscious percept. And let's talk quickly about top-down feedback. This feed-forward framework tells us a lot about how neuronal tuning properties changes across different stages of visual processing in the brain. But feedback is massive in the brain in terms of the synaptic weight, and its functions are still being elucidated. So, for example, even in primary visual cortex, we know from anatomical studies, less than 1% of the synapses come from the feed-forward pathway from the visual part of the thalamus. And the vast majority of the synapses, 99%, are accounted for by intrinsic horizontal connections between V1 neurons or by top-down feedback connections from higher order regions sending information back to V1. And our GN is the same. Less than 20% of the synapses are accounted for by the feed-forward pathway. The vast majority of the synapses come from the top-down feedback from V1. And we think that this top-down feedback performs a variety of functions, not just attention that is widely recognized, but also predictions and perceptual template. So in this paradigm I mentioned earlier, let me show you two examples where we've looked into top-down function. So one is that during these false perceptions where people say, I see a meaningful content, meaningful object, a face or a dog, when the stimulus input is these meaningless noise patterns. We found that if we contrast the real image trials with the scrambled image trials, the scrambled images elicit stronger recruitment of higher order brain regions. So these two blue regions in prefrontal cortex are activated even more than real image trials, and the default mode regions shown in orange are deactivated even more, than real image trials. And this is not just the task difficulty effect, because we could decode the content of that false perception in the scrambled image trials in these regions. For example, here, in these two blue regions, the gray bars shows our ability to decode the category of their false perception content, and it almost matches what you see for the real images when they are being recognized. And when the real images are not recognized, we could not decode the category of those object images. So suggesting that these regions reflect perceptual content, and in the false perception case, they may be sending this top-down template to sensory regions, which carries kind of these top-down regions guess about what may be out there. And the second example I want to give about top-down feedback is about how our lifelong experiences create priors in the brain that influences perception. So this is a 2D picture, but you see these ones as concave and this one as convex, because we have these powerful priors in our brains about how light comes from above. We can't override that prior because it's lifelong experience. And it turns out that in this bistable perception paradigm I mentioned earlier, even though this nickel cube is physically symmetric, people tend to perceive this view more often than this view. And that is because throughout our lives, we see boxes lying on the floors rather than hanging in the air. And this bias is highly stable within an individual. And using intracranial EEG, which is a direct cortical activity measurements in neurosurgical patients, we were able to ask what is the information flow pattern in the brain when people perceive the more preferred view that is consistent with your lifelong knowledge, or the non-preferred view that's inconsistent with your lifelong knowledge. And we use a method borrowed from neuroscientists, borrowed from econometrics, called Granger causality, to ask directed information flow between brain regions. So the idea is that if I can use the past information from region X to help me predict the future of region Y, there may be information flow from X to Y. So what we found was that in the ventral visual pathway between occipital and temporal cortices, when people perceive the non-preferred view that is inconsistent with your lifelong knowledge, there is a drastically enhanced bottom-up connectivity. And when people perceive the preferred view that is consistent with your lifelong knowledge, there is much more strengthened top-down feedback. And so we were able to capture this with parsimonious computational model, which starts with an attractor network model, also called double well model in physics, where you have two groups of neurons that mutually inhibit each other. They compete with each other, and each one has adaptation, so this activity goes down over time. And it's well known that this attractor network model can model the bistable perception behavior, but by itself, it doesn't model this bias that we observe behaviorally. So when we extended the model to be hierarchical and incorporated top-down bias that strengthened one of the pathways, we could reproduce both the behavior pattern as well as this neurophysiological observation. To put simply, prior knowledge from lifelong experiences can be stored higher up in the brain and influence perception through top-down bias to bias one of the two interpretations in this paradigm. So in the last part, I'd like to share with you our work on one-shot perceptual learning. One-shot perceptual learning refers to the fact that a single experience can drastically influence our conscious perception. So it was initially famously illustrated by this picture, which was created by the British psychologist Richard Gregory in the early 1970s. So this picture looks like meaningless pattern of black ink blobs on a white piece of paper if you've never seen it before. But hidden in this picture, there is a dog. Here's a dog's head sniffing the ground. Here's the body. And the four limbs are here. And it's next to a tree, the bottom of a tree. It's a very famous picture in psychology because it illustrates the principle that once you recognize that dog, every time you see this picture from now on, that dog jumps out of the picture at you. The perceptual completion process is automatic and fast. Gregory summarized this effect very well in this sentence, perception is solving puzzles. It also illustrates the principle that the human brain is very data efficient and energy efficient, and is able to have this one-shot learning without catastrophic forgetting. And that contrasts with modern day AIs being data hungry and energy hungry. And this is something that we are very interested in from a computational perspective. So some years ago, my lab developed a pipeline to automatically create many pictures such as this. And this is one of the pictures we created. If it's your first time seeing it, it probably is very difficult to recognize, looks like a meaningless pattern. But now if I show you this picture, many of you will see this picture as a dog. There's a dog hidden in it. And if this one doesn't work for you, a different picture will. Here's another example. And here's the original picture with a chair in the middle. And now you might see this picture as a chair. This effect is very robust. People's accuracy in recognizing these degraded pictures are low to begin with and high after that one-shot learning experience. And the learning effect is long-lasting. If you bring somebody back to the lab many months later, that learning is still there, that memory is still there. So what's the neural mechanism for one-shot perceptual learning? A default hypothesis here is hippocampus, which is arguably the most famous brain structure. It's very famous because it's well known to support fast learning. So long-term memory can be categorized into declarative memory, which really meant memory that we can consciously recall, and non-declarative memory, which is, memory that we cannot consciously, it's a little jumpy, is that we cannot consciously recall. And the hippocampus is well known for supporting one-shot declarative memory, including episodic memory about events and semantic memory about facts. And the cortex and the striatum are brain structures that's well known to support non-declarative memory. The most famous being procedural learning or motor skill learning, like learning to ride a bicycle or play the piano. And I was very fortunate to be able to team up with Larry Squire, one of the pioneers in memory research, to test patients with amnesia. So they have a severe loss of episodic memory due to bilateral hippocampal lesions. And we tested them on this task. So these are the brain scans of the 5 patients we tested. On the right is the scan from a control participant. You see the hippocampus is outlined in the controls. And where the hippocampi used to be in the patients, now you see these black holes. And based on the volume loss in the MRI, it was estimated that there was severe to total loss of hippocampal neurons, and all patients had classic amnesia. So we tested patients and age-matched controls on our one-shot learning task, and we found that, in fact, patients were completely intact on this task. So the first bar is the identification accuracy for the degraded picture before seeing the grayscale picture. And the second to the fourth bars are 1 day after seeing the grayscale picture, 7 days after, 5 months after. You see that patients do as well as controls. The white dot is GP, who had lesions of both bilateral medial temporal lobe and severe amnesia, and was able to learn this one-shot perceptual learning task just as well. So this suggests that hippocampus is not needed for one-shot perceptual learning, but the neocortex supports it. And there was a beautiful double dissociation here, because if we change the task from naming the object in the picture, which is what this one-shot perceptual learning task is about, to have you seen this picture before, which is a classic memory task, then patients are suddenly very impaired. So the gray bars are patients, the black bars are controls. The left one are naming tasks, the right ones are from the memory task. So how does the neocortex support one-shot perceptual learning? We initially looked into this by neuroimaging. This was another whole brain 7T fMRI study, where we found that across all of these brain regions, if we compare the neural activity pattern driven by the degraded picture to the matching grayscale picture, we found that in all these brain regions, the degraded picture triggers a more similar activity pattern to the matching grayscale picture after learning. But this doesn't tell us where that learning is happening in the brain, because it doesn't make sense for the brain to store a copy of the memory in all of these brain regions. Rather, what happens is more likely to be like this, where the synaptic connectivity, synaptic connections between neurons in one of the regions is modified, are modified to store the memory. And once that memory is reactivated, triggered by the matching degraded picture, post-learning, once it's reactivated, it can have widespread effect on connected brain regions. So in this recent study, we used 3 investigations to try to narrow down where that learning is happening in the brain, where the plasticity is stored. First, with psychophysics, we asked what kind of information is stored in the prior knowledge. And second, with intracranial EEG, we asked which brain region has the earliest onset activity difference before versus after learning. The idea is that the region with the earliest onset is more likely to store the memory, because that memory has to be reactivated before it can have this network impact. And lastly, we tried to build a neural network model to capture the behavioral effect in humans. And let me just show you the intracranial EEG results first. We used two different analyses here. The first one was just looking at neural activation to pre-disambiguation degraded picture shown in blue, post-disambiguation degraded picture shown in orange, and gray scale pictures, the original pictures shown in gray. And what you can see is that in high-level visual cortex, the human homologue of IT cortex, there is the earliest difference between pre and post images, shown by the difference between the blue and orange lines and the black bars at the top. And the bottom analysis was more fine-grained. We asked for each degraded picture, how similar is the neural activity to the matching original grayscale picture. And you see that in the same brain region, that similarity shoots up in the post-learning stage. So the neural activity becomes similar to the matching prior picture. And this effect happens earlier in high-level visual cortex and early visual cortex, suggesting a top-down feedback effect here. And my collaborator, Eric Allman, and his student, Chris Liu, built this wonderful model where we augmented a vision transformer model, DanoV2, with a top-down prior storage module. This top-down module learns information and accumulates it across a visual sequence and use the top-down conditioning to influence the visual processing in the transformer network. And the model was able to capture the one-shot learning behavior exhibited by human subjects, as well as the error pattern by humans. In other words, when humans learn, the model learns. When humans are not able to learn an image, the model also fails. So the bottom here are the similarity between two different model instantiations, between two different human subjects, and between our model and human subjects. So the similarity between our model and human subjects also almost matches up to inter-individual human subject similarity. So to summarize, the visual awareness in the brain is generated through an interaction between bottom-up and top-down brain activity, and an interaction between spontaneous activity and evoked activity. It's the brain's interpretation of the sensory input in the context of past memories. I'd like to leave you with two parting thoughts. One is that I talked extensively about visual perception today, but conscious versus unconscious is an organizing dimension of other mental functions. For example, motor functions could be volitional, conscious, or automatic or reflexive. Emotions have a conscious component, what you feel, the felt emotions, and the unconscious component are the somatic markers that change with emotional reactions, which may occur without your felt emotions. Learning and memory, as I mentioned, could be conscious and explicit, or unconscious and implicit. So really implicit means the same thing as unconscious, and explicit means the same thing as conscious. And thinking, we know, has two different modes. We can think consciously, deliberate, serial, deductive. Or we can think unconsciously, where the brain is doing all the work in the background and a solution pops up in our head as a cognitive insight. So all of these phenomena are very important to study for understanding conscious awareness and how it's implemented by the brain. And lastly, if you're interested, I recommend this article that I wrote a couple of years ago to try to map out a general framework for how empirically in neuroscience we could approach conscious awareness, which is to take the state space view and try to identify axes, different axes that matter to different contents or states of consciousness. A benefit of this framework is that it unites states of consciousness research with content of consciousness research and allows us to ask questions such as how experiences in different sensory modalities interact with each other. So I'd like to close by thanking my collaborators, especially the names in yellow are people who were really essential for all the work I told you about today, my funding sources, and thank you for having me here.
Thank you very much, Biyu, for that fascinating talk. Just in the interests of this series, I'd like to ask two very quick questions. What do you think is the nature of reality? Is it something external that the brain is trying to perceive in certain way? And another quick question is, what is exactly the role of the brain? Because some people, I mean, you might have heard of these arguments about hydrocephalus, and really, is there a need for a brain in order for consciousness to exist? Right? So, what are your thoughts? Nature of reality and also consciousness and the role of brain.
Okay, the second question, I'll start with that. It's a little bit easier to answer. Um, so people with hydrocephalus have much smaller neocortex, but they typically retain intact subcortical circuits. So in this review, I mentioned at the very end from that I wrote in 2023, I talked about the role of cortex versus subcortical circuit. And there has been a long tradition of thinking in our field, in the field of consciousness, about the roles of subcortical circuits in conscious awareness. And I think the consensus today is that those circuits contribute to conscious awareness. And there are powerful arguments from clinical observations that people with hydrocephalus have conscious awareness that is mainly supported by their remaining brain circuits, that is the subcortical circuits. And so what I think is that, and also, subcortical circuits are much more ancient evolutionarily than the neocortex. It's what we share with a lot of other animals. So my overarching idea here is, which I talk about in that review, is that the subcortical circuits are able to support relatively rudimentary awareness, maybe not as richly differentiated content. And the neocortex enriches that content to be much richer and multidimensional, etc. And your first question about reality, what is the nature of reality? I'm not a philosopher, say that at the outset. There is definitely a physical reality out there, obviously. The question is, I think the scientific question that's interesting to us neuroscientists is how does the brain generate its own reality, which obviously can be decoupled from the physical reality, such as when we dream. And there was this really nice experience where 50 or some more years ago, where when people are in prolonged sensory deprivation, the brain starts to hallucinate. So and kind of come up with its own reality. So it's like most of the time, what we experience is constrained by external sensory input and is in touch with physical reality, but sometimes that process can go awry. And in my lab, we have been very interested in visual hallucinations, which is a topic that's directly we got into it because of all of our basic science studies about conscious perception. So we are very interested in how the brain generates hallucinations in the visual domain. And that is, an example where there is this altered reality that the brain is living in that's uncoupled from sensory input.
Thank you for that insight. I'd like to quickly go on to a question, actually, maybe three questions asked by Stuart Hamroff, who is one of the other organizers of this series. So he asks, of course, he says, great talk, and thank you for this very insightful talk. Are you considering deeper, faster, denser cytoskeleton dynamics in ongoing activity? And there are other parts to this, but I'll ask you later. Yeah, so deeper, faster, denser cytoskeletal dynamics in ongoing activity. Are you considering the role of that?
Okay. Hi, Stuart. Thanks for being here. The short answer is no, because the spontaneous dynamics we are talking about here are um, the neurochemical and neurotransmitter related dynamics that mostly reflect interactions between neurons. And they get picked up by our techniques because they manifest as either subcellular membrane potential fluctuations or, most importantly, spiking activity changes over time. And I'm not aware of solid empirical evidence connecting those neuronal dynamics with cytoskeleton changes in the cytoskeleton.
Okay, so the second part is, are you considering backward time effects in pre-stimulus activity? And I know Stuart has been discussing this for a few years now. Have you considered that backward time effects? Of course, it's very bizarre, but could something like that be occurring? Perhaps even against
If
physics
you
as
could
we know it.
define the backward time effect for me, like because different people could mean different things with that phrase.
Well, you showed the graphs on, you can predict the activity before you show the stimulus. I guess that's what he's referring to. So could there be backward effects time or backward time effects going on
I
in
just
the,
don't know what backward time effect means here.
meaning something which is supposed to be happening in the future, you can already notice its effects in the past, right?
Okay, and there's nothing mysterious here, like of what we are observing. So, what we are observing here is that the brain state prior to the stimulus input has an impact on how that brain, how the brain processes that stimulus. And that is very, very well known, in fact, in neuroscience. That just think about it from a single neuron's perspective, right? Like its firing rates is going to influence how it processes, how it reacts to synaptic input. So, and then that effect, is really a multi-layered effect, will be observed at a local population level, a regional level, a large-scale brain network level. The brain is a dynamical system, and the property of dynamical systems is that its state at one time influences how it's going to behave in future. So it's there's nothing mysterious here.
Okay, so no backward causation or something, right?
Exactly.
Right. Okay. And on to Stuart's third part, I guess that's his favorite question is memory. Have you considered memory encoding and one-shot learning in microtubules by calcium M key? Yes, CAM key, basically.
No, I have not.
Okay. So moving on to some of the other questions. Here's another one. Does a one-shot sensory exposure remove or greatly reduce the feeling of novelty because the brain encodes a memory of the encounter, even without conscious awareness? And does it matter to the brain where the first encounter occurred?
Sorry, what was the last part? Does it
Does
matter to
it
the brain what?
matter to the brain where the first encounter occurred?
Um, so in terms of a first encounter changing how the brain reacts to the second encounter, that's a very well documented neuroscientific and observation, and also in the behavioral psychological literature. Like the novelty will reduce. There is also this repetition suppression literature in neuroscience where the brain's response to the same stimulus presented second time will be smaller. And there was also this question about what happens if you're not aware of the first stimulus. And there is this wonderful behavioral literature called subliminal priming, which really provides very strong evidence that the brain can process sensory input unconsciously. It's basically showing that a stimulus that is not consciously perceived can change somebody's behavior or brain response to the same stimulus or related stimulus later on. So even when it's not consciously perceived, it can leave a trace in the brain that changes your reaction to the same stimulus presented later or related stimulus. Um, And there was a question of does it matter where this is encountered? I assume it was about where in the brain this happens. Um, so I think the short answer is probably yes, that it matters if it's a sensory region or associative region. One of this question, this may be a side track from the question I was being asked. But if you read our paper that came out this year about one-shot perceptual learning, in the discussion section, we talk about the relation between this phenomenon and priming. So in that memory diagram, memory classification picture I showed, that priming, which is under the same bracket as procedural learning. The priming is a well-documented effect that's supported by the neocortex. And we think our hippocampal lesion study essentially places one-shot perceptual learning under the phenomenon of priming. But it illustrates how robust, dramatic, and long-lasting priming effects can be, which is not something that was previously known. Yeah, so I guess that's all I have to say about that.
Thanks. Here's a question from Saul Markman, who asks, do you think that long-term priors versus recent experience are incorporated differently as priors that bias perception? So he gives a couple of examples of long-term priors is intuitive physics, for example, and an example of recent experience is watching a show with a certain visual style.
Yes, I like that question a lot. Very good question. Yes, the answer is yes. So I think they could very well be different. So in fact, I think mistake that's often made is to think that the brain has one type of prior and the same mechanism will explain different types of priors, which I think most working neuroscientists in my field recognize is too simplistic. There are different types of priors. There are priors from lifelong knowledge, priors from something you told someone about, like, a very popular paradigm is you're telling the subject on the next trial, the stimulus will most likely be a high pitch tone or a low pitch tone. And, that's kind of a very short-term prior that only exists now, in the context of that task. There are like this example that you just gave, a visual experience that happened recently or long ago. So there are different types of priors. And as good empirical scientists, our job is to decipher the brain mechanism for the different types of priors and how they impact processing. For example, long-term priors, I talk about top-down mechanism for encoding long-term priors, but it doesn't have to be only top-down mechanism. Bottom-up mechanisms could also implement long-term priors. So it's an interesting question for the field going forward, when top-down is brought in and when it's bottom-up.
Thank you. We'll take a couple of very quick questions. Here's another one. How do the top-down versus bottom-up findings map onto the schizophrenia literature and mechanisms of drugs that cause hallucinations?
Very good question. So I don't have a lot to say here about the drugs that cause hallucinations because that hasn't been my own research topic. Also, I've watched that field with a lot of fascination and interest. What I do have to say is about clinical hallucinations in schizophrenic patients or neurological patients who experience hallucinations. There are multiple strands of evidence suggesting that the top-down may be overweighted in those cases as compared to a neurotypical brain. The idea is that hallucinations may partly be triggered by the brain relying too much on top-downs that are not entirely appropriately informed by the bottom-up sensory input. Yes, I think probably both malfunctioned interaction between bottom up and top down, and malfunctioned or maladaptive spontaneous activity could play a role in generating hallucinations. And there is a lot of good empirical work that still needs to be done in that domain.
Thanks. We'll end with a last philosophically inclined question, which sort of resonates with some of the initial point that I had asked. But this question is from Arnaud Delorme. Probably know him. Is consciousness an emergent property of the brain, as per your understanding, or maybe as per your belief? And is qualia an illusion created by the brain, or could there be more to qualia? And I'd perhaps add a little more to this on I mean, what is the experiencer? Is it the brain? Should it be something different? And this ties back to one of my favorite questions is if we were to find life elsewhere or search for life elsewhere in our universe, would we be looking for neurons, neuron-like material characteristics, or could it be some completely different kind of consciousness-based life?
Hmm. Um, the experiencer is the brain. Since I'm a monoist, the so your earlier question was about whether consciousness is an illusion. I don't think so. And I think the question, so you're basically asking about the hard problem of consciousness, I guess. I think the same problem applies to pretty much all the other mental phenomena we study in cognitive neuroscience. So it could be working memory, could be emotion, felt emotion, and could be attention, voluntary attention, for example. So in cognitive neuroscience, we have a set of terminologies describing the mental phenomenon, and which we measure through behavior. And we have the neuroscientific terminology describing the brain phenomenon. And I really think those are two sides of the same coin. And I don't think there is a huge gulf that we cannot bridge. So the last part of your question was about the substrate, if I got that correct. Like what kind of substrate can support consciousness? I am open-minded about this. I don't think it must be neurons as we find them in the brain here on Earth. I think it could potentially be implemented by a different medium. Although I have to confess that I share the skepticism of people like Ned Block about the ability for modern day, current day digital computers to support conscious awareness. I'm not a philosopher, so I'm not going to mount a philosophical arguments here, but that's where my intuitions lie. And I think neuromorphic computing maybe physical medium that's closer to the brain, but doesn't have to be exactly the same as the biological brain could support awareness.
Good response, Biyu. Before we close, I'd like to mention that the next colloquium in this Consciousness and Reality series, which will be the final one for this academic year, will be held in June, and that will be delivered by Nobel laureate Sir Roger Penrose. And again, I would like to thank you, Biyu, for that fantastic and thought-provoking talk. I'd like to thank all organizers and participants who made this event happen. Thank you very much and have a great day. Take care now.
Thank you for having me.
Thanks.